Uh, okay. For today, we have a great opportunity from Professor Sarawut Kitpakdi from Department of Biochemistry, Faculty of Science, Mahidol University, to give you a very special lecture because he is one of the expert in cancer metabolism. He has many publication, a lot of publication on the topic of cancer metabolism, and he will give you the basic knowledge and the advanced knowledge about the research work from his experience. Okay, so Professor Sarawut, please.
Yes, thanks very much, Ajahn Piti, for your introduction. Can you hear me okay?
I can hear you quite well.
Okay, thank you. Okay, let's see. Yeah, what I like to do today is I want to share my experience on the metabolism of cancer cells. So I hope by the end of the lecture, you had a better idea of what's going on in the cancer cells, why the cell proliferate so fast, and why there's a certain pathway are essential for survival of the cancer cells, and what metabolite or specific pathway are actually selectively activated, and that's promote the progression of cancer and also the chemotherapy as well. So, okay, so you also know that the hallmark of the cancer cells, they are reprogramming of several biological process occurring during the oncogenesis. And on the right hand side, we have what we call the hallmarks, or the views of the cancer cells characteristic. I'm sure you all appreciate that the most obvious change or biological change happening in cancer cells are the invasion and metastasis, because these two biological process are the play essential role in the progression of cancer, and that's of course leading to the death in many patients. So we all know that invasion and metastasis are the key important of cancer cells that enable them to spread to the distal organs and other tissues. And of course, there is been long known that they sustaining the proliferative signal, otherwise known as the oncogenic signalings, and that's also a primary event that cancer cell really adjust the biological process, making them keep dividing all the times. And there's also the evasion of growth suppressor, and this is of course the unlike quiescent cell or differentiated cells where the cell stop dividing, but in cancer cell there is a signal to invade growth suppression. There's of course there's the evasion of immune destruction. Somehow the immune cell wouldn't work that well to eliminate cancer cells. There's the enhance the replicative immortalities on the right hand side. I was trying to get my pointer to work. Hang on. Okay. Everyone see it, right? Of course, there's about 9 biological process that's been going on, and oncologists known it for many years, but until in the last 10 years, everyone appreciate that not only the biological process I talk about, there's also the reprogramming of energy metabolisms in cancer cells. So this is one of the important hallmark which enable the cancer cell to increase the biomass production. So as you know it from the left-hand side, there's a genetics mutation contribute to the oncogenic transformation. As you know that the external factors or in genetics factors contribute to the oncogenic signals. And when the signal is activated, and that's in turn the um, uh, reprogram the metabolisms. That's what we know as the metabolic adaptations that enable the the transformed cell to become the uh, malignant cell with the increase of biomass production. So you think about the proliferative cell or cancer cells, where the oncogenic signaling is become activated. That's going to be the right hand side of this diagram. So you compare it with the normal cells when the cells receive the appropriate mitogenic signaling, they undergo cell cycles and divide it. But then there's the signal to stop it. So means that the normal cell can keep only the certain round of mitosis, whereas on the right hand side, there's the amplification of oncogenic signaling, and that allow the cell to keep dividing so many generations. Um, this is of course resulting the formation of the tumor mass with the massive number of new cells. So you think about in terms of metabolic stress, this you know excessive oncogenic amplifications force the cell to increase the biomass, cell membranes, nucleus, and other structural component of the cells. And this, of course, if the cell not cancer cell not doing anything, that's going to be bad for them. So means that under this excessive oncogenic signaling activations, you get massive biomass productions, and that's biomass. What is it? That's going to be the structural component of the cancer cells. Means that the new cell will require massive amount of cellular components, as I listed on the right-hand side. Those structural or cellular components include the nucleic acid. Of course, this is um, the component of nucleic acid or genetic materials, the membrane lipids, the organelles. Of course, new cell need to have organelles. New cell or cancer cells still need a lot of structural protein and enzymes to be able to maintain their biological activities. So another thing also, you think about the reprogramming of the metabolism in cell, perhaps attribute to the high requirement of the ATP. You all know that ATP is the chemical energy for living cells required to drive several biological process. What I have on the right-hand side is the biological process happening in living cells, but specifically for cancer cells. So of course, the ATP is needed to drive proliferation, stemness, and collage. independence, cell migration, metastasis, cell invasion, and about all sorts of things. So that's going to be to drive the non-metabolic process. So that's explain why if the cancer cell do not reprogram metabolism in the way that they produce enough ATP, they're not going to be able to survive. I think that's the main idea of the metabolic readjustment or metabolic reprogramming. So to increase biomass in response to mitogenic signaling, and also to serve enough ATP for the non-metabolic and metabolic processes. The idea of the metabolic reprogramming in cancer cell perhaps was pioneered by early scientists back in 19th centuries. This pioneers person named Professor Otto Warburg, who was later on awarded the Nobel Prize in discovering the metabolic reprogramming in cancer cell. So what the Warburg observed was that the cancer cell perhaps differ from normal cell in the way that they consume large amount of glucose. And how much we're talking about? We're talking about 10 times more than the normal cells. And why the cancer cell consume large amount of glucose? This is because most of this glucose are metabolized through the lactic acid, production, and that's again rate of lactate production, which 100 times more than normal cells. And you can imagine this will create a very, very acidic environment around the cancer cells, which is not happening in the normal cells. So what does it mean when you have a very far, rich rate of lactic acid production? Means that cancer cell very likely to utilize the glycolysis rather than the oxidative phosphorylation to generate ATP. So I hope you all recall about the OXPHOS. This is the method of ATP production by using the mitochondria to oxidize the electron acceptor molecules, which are basically an ADH and FADH to produce ATP. And unlike cancer cell, they tend to use the glycolysis, which, if you remember, the glycolytic pathway only produce a few couple molecules of ATP from one molecules of glucose. And at that time, Warburg thought that his thought was that reason for this was because the most cancer cell harvest the defective mitochondria that's limited the ATP production by oxidative phosphorylation. So you can imagine if we have no mitochondria, the oxidative route of glucose production will only be through the glycolysis. And all of this for observation led the Warburg to think that cancer cell can then only live under an aerobic condition, because around cancer cell there's a lot of lactic acid surround the cell, so they thought that cancer cell can only live under acidic condition, but will soon die under aerobic or alkaline conditions. That's at that time people thought this hypothesis that all of these 5 observations are correct, but later on there's only observation number 1, 2, and 3 are proven to be true. In fact, it's well known that the mitochondria of cancer cell, in fact, is not defect, but it's rather the metabolism in the mitochondria is rather reprogrammed to suit to support survival of cancer cells. Okay, and of course, now if you try to kill the cancer cell by putting them in alkaline condition, I don't think there's I don't think not only cancer cell will die, but normal cell will also die. So, okay, the as I said, one of the classic observation of Wolfsburg is that the cancer cell uh, 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 oxidize glucose through the glycolysis, and that's well supported by the observation this day. In fact, those of you who work on cancer cells, you will notice that when you culture cancer cell in the flask with the well-known media like DMEM or RPMI, those sort of media often supplemented with the phenol red, which is the uh, pH indicators. So you notice that the color of the media of the fresh media is pretty much sort of red, pink color, and that's because the pH is around about 7.8. So, but when you keep the culture growing for a certain time, you start to see the change of the media color from pink become orange and even to the yellow color, and that's because the pH, of course, in the media it changes attributing to the secretion of lactic acids from cancer cell, and that's support Wolfsburg hypothesis quite well, because the phenol red in the media, when there's the acid environment, the molecules become protonated and eventually to the yellow color, and that's indicate that cancer cells somehow secrete the acid and make the environment around cancer cell is rather acidic. Uh-huh. So what does it mean? What does this mean comparing with the um, proliferating tissues? In fact, if you look at the tumor cell, which shown on the right-hand side of this slide, comparing with the proliferative tissues, means that it's still considered to be normal tissue, but still divided. So if you grow proliferative cells or tumor cells in the presence of or absence of oxygen, you see the difference is that tumor cell, no matter oxygen is there or not, they will prefer to oxidize glucose and produce lactate pretty much 85%. Only 5% going to the mitochondria. So that's similar to proliferative tissue as well. But then proliferative tissue depend on oxygen. So if the oxygen is there, glucose will be metabolized through the oxidative phosphorylation. So for example, the skeletal muscle as one of the examples, when the oxygen is not there, metabolic pathways switch from aerobic to anaerobic, depend on oxygen. But for tumor cell, even though you have 100% of oxygen, this is very true when you grow the tumor cell in incubators. That's the concentration of oxygen in the oven is really high, but tumor cells still metabolize glucose through the glycolysis and produce lactate, even though oxygen is a lot, you know. That's the hallmark, the difference between cancer cells or proliferating cell, which is shown on the right-hand side of this slide. And, most of the primary metabolic pathway is the glycolysis and very low rate of oxidative phosphorylation or mitochondria metabolisms. And there's also the high rate of biosynthesis happening in proliferating or cancer cells. On the opposite way, which is shown on the left-hand side, the highly specialized cells or the differentiated cell, for example, neurons, endocrine cells or skeletal muscle cells, also well differentiated. They would metabolize glucose through the oxidative phosphorylation. Otherwise, what you see on the left-hand side, totally opposite to the proliferating cells. So that's pretty much the major difference between the the differentiated cell or quiescent cells or the cell that already stopped division, or in other words, the cell that already enter in G naught cell cycles. Whereas proliferate cells keep re-enter the cell cycles all the time. So you think about why the cancer cell would prefer the glycolysis to produce ATP. As you know that the pathway that produce ATP coming from the oxidation of the electron acceptor molecules, which is NADH. If you remember, NADH are the product of glycolysis. What else? TCA cycles that generate a large amount of NADH. And to be able for the cell to extract the ATP, cell needs to pass the NADH through the mitochondria membranes so that the oxidation can be occur by pulling out the electron from NADH and pass through the respiratory chain to produce ATP. That's very effective pathway to generate the ATP through the oxidation of electron acceptor. So unlike the fermentation or glycolysis, where the NADH reoxidized through the oxidation of pyruvate to lactate. So this way, you don't actually produce a lot of ATP that much. But then ATP only produced from the payoff phase of the glycolysis. So this is what I just told you before. You look at the overall pathway of the differentiated cells. When the cell uptake the glucose and metabolize, you end up with the glycolysis, which produce only 2 molecules of ATP. Then NADH generate from glycolysis pass through the electron transport chain, and also the pyruvate, which is the final product of glycolysis, going into mitochondria to be converted to the acetyl-CoA molecules, and that's enter TCA cycles. And the hallmark of TCA cycle, as you all know from the basic metabolisms, TCA cycle also produce a large amount of NADH and also FADH, and the combination of cytosolic and the ADH from glycolysis and TCA cycles pass on through the respiratory chain to produce ATP. And that's why in the differentiated cells, after catabolism of glucose, you produce up to 36 molecules of ATP. This is unlike cancer cell, which I already mentioned. There's the determination of the flow of the pyruvate into the mitochondria. So in other words, in the cancer cell, glucose catabolism will only finish at in the blue shaded here, means that we only get 2 molecules of ATP from glucose catabolism. Now, what would be the benefit or advantage of not having the rest of glucose catabolism? In the green box here, activated. Perhaps the answer is lying to the last 2 lines of this slide. Now you think about the turnover rate of ATP synthesis from metabolism of glucose. Oxidative phosphorylation, you notice that this, to be able to get the ATP production from mitochondria, you require quite a number of reactions starting from glycolysis, Krebs cycles, and then pass on the NADH to the respiratory chain on the mitochondria. That's involved more than 25 biochemical reaction. Now, 25 means that can take very long time to produce ATP. In fact, when the biochemist or metabolic biochemist measuring the ATP production rate from glucose catabolism, they found that it requires up to 5 minutes to produce ATP from glucose. That seems to be a certain time point, whereas the ATP production from glycolysis only take about 10 to 30 seconds. So you notice that that's about 10 times faster than producing ATP by the oxidative phosphorylation. The reason why it's much shorter is because you count the number of glycolytic pathway. There's only 10 reactions here. That's why it requires shorter time comparing with the rest, which you need to pass on the pyruvate to mitochondria, and then acetyl-CoA production have to be occurring afterwards. Then acetyl-CoA molecules pass on to the TCA cycles, and TCA cycle also require up to 8 chemical reaction. So that is that up. That's why ATP production somehow delay or slower than what happening with the glycolysis. Now, some of you might argue that okay, ATP synthesis rate is faster, but would that serve enough ATP? The answer is cancer cell can in fact speed up the glycolysis rate about 100 times higher in cancer cell. This is to compensate for the ATP production. In other words, you might think glycolysis is quite a poor ATP production pathway. How the cancer cell cope with that? Answer is they can speed up or they upregulate the glycolysis pathway faster, so that, and in the end, the total ATP really similar to what you get from oxidative phosphorylation anyway. So that's the explanation why glycolysis is sort of benefit for cancer cell in terms of ATP production. It is faster ATP production process, and cell can upregulate or increase the glycolysis to operate many rounds, so that in the end you produce the same or equivalent amount of ATP generated by the oxidative phosphorylations. Okay, what else would be the benefit of relying on the glycolysis or lactic acid production rather than using the oxygen to oxidize glucose? This is perhaps true for the solid tumor. You know what solid tumor is? So solid tumor is the very dense mass of tumor which often result from very rapid proliferation. So you think about this is what I have the right hand side. This is the kind of the uh, schematic drawing of the solid tumor cells. So we start off with the different layer of cancer cells growing close to the vascular structure or blood vessels. So which is red color here. This is blood vessel. As you all know that the cell that grow well beneath the blood vessel who have well supplied with glucose and oxygens, because you know that hemoglobin is the essential molecule that will carry oxygens to the cells. And not only that, but blood vessel also supply nutrients, specifically glucose molecules. In other words, the cell which grow close to the blood vessel will be able to access to the glucose and oxygen quite well. So this will form the layer of cells we call the aerobic cells. Okay. You look at the metabolism of aerobic cell, it is pretty much rely on glucose and oxygen. So if the oxygen is enough, aerobic cell will be fine. You know, they grow nicely and form very loose, loose structure of the tissues here. But deep down, beyond this point, where the flow of oxygen is become limited, you're going to have the layer of cell which suffer from lacking of oxygen. We call the hypoxic layer. Okay, and that's we call the hypoxic viable cell, means that this population of cancer cell lack of oxygen but still able to survive, but not as good as the aerobic layer of cells. Okay, but the most distal group of cells, they not live well at all. In fact, they become necrosis because they not enough oxygen and glucose to flow deep down there. So that's create the necrotic population of cancer cell here. Okay, now you see that if all three layer of cancer cell rely on oxygen metabolisms, what would be happening? You're gonna end, most of cancer cell will end up with hypoxic cell or necrotic cell, right? Because the deep down here there will not be enough oxygen and glucose. That's reason that the explanation why most cancer cell would prefer an aerobic metabolism or glycolysis to oxidize glucose, because that will make the cell less risky to oxygen deprivations. You look at the oxygen concentration across the just beyond the vascular structure or blood vessel. So here we have the hemoglobin, red blood cell that carry oxygen. So often the oxygen concentration immediately beneath the blood vessel will be very high. It's about 12% of oxygen, whereas deep down to the tumor tissues, oxygen concentration is limited to 1%, and this is because the blood vessel is not well supply to the deeper area of the tissues. And if there's the tumor is formed right underneath blood vessel, you can imagine the area which proliferate deep down the blood vessel, lack of oxygen, make them immortalized. Sorry, make them die in the end. So by switching to the anaerobic or glycolysis, this would allow the solid tumor to depend much less on oxygen. And that's why glycolysis is very important for them to be able to metabolize the glucose, because the glycolysis doesn't require oxygen. Okay, that's allow the cancer cell to survive even though the blood supply is not deep enough. Okay, and the glycolysis not only in terms of provide ATP production at the much faster rate than oxphos, and also the cancer cell can tolerate to the oxygen deprivation condition more. What we also know is that the benefit of having very active glycolysis is that you favors the biosynthetic pathway. As you can see from this slide, start off with the catabolism of glucose through the glycolysis, and you produce a pyruvate. In the well-differentiated cell, as I mentioned before, pyruvate further metabolized to acetyl-CoA, and the acetyl-CoA molecules enter into the TCA cycles. And that's known for the differentiated cell or normal cells. So being the tumor cell, there's the signal which will inhibit the entering of pyruvate into acetyl-CoA enzyme A in the mitochondria, and that force the pyruvate to be converted to other metabolites. As you can see here, more than 85% of pyruvate, in fact, diverting or converting to lactate molecules, as I show on the left-hand side. Now, imagine if the reaction downstream, which is the conversion of pyruvate to acetyl-CoA molecule, is inhibited in cancer cell, what would be happening? Means that you end up with the accumulation of pyruvate, right? This is the basic concept of enzyme kinetics, right? If you block this reaction, which I put the cross mark here, indicating that the conversion of pyruvate to acetyl-CoA is 100% inhibited, means that you're gonna accumulate the pyruvate into the cell. Okay, now when you accumulate the substrate here, means that pyruvate have to go somewhere else. It cannot pass down to the acetyl-CoA, of course, because this point is inhibited. Means that pyruvate can either go to lactic acid productions or going into the alanine productions, and also can flow upstream to the reverse direction of glycolysis. Means that in the cancer cell, there will be the higher concentration of glycolytic intermediates. For example, the PEP, glycol 3-phosphate molecules, fructose 6-phosphate, glucose 6-phosphate molecules here along the pathway as a result of inhibition of the pyruvate dehydrogenase reaction, as I put the mark here. means that you automatically enhance the accumulation of glycolytic intermediates. Where would the glycolytic intermediates go then after the concentration are elevated or upregulated? Means that some of these glycolytic pathway have to go to other reactions, including the pentose phosphate pathway. As if you still recall, pentose phosphate pathway is the minor root of glucose metabolism. This pathway PPP in fact essential to produce the ribose 5-phosphate, which is the backbone of nucleotide. Phosphoglycerate GP here is the precursor for serine biosynthesis. And you know that serine is one of amino acids required to modify the phospholipids head. Phospholipid comprise of lipids, phospholipids, and the amino acid. And serine is one of amino acid which is required to modify the head structure of phospholipids as the component of cell membranes. Amino acid also required for as the backbone of polypeptide. So you see, and also glycerol molecules in fact derive from the HAP, and that glycerol is the essential alcohol which is required to be esterified with free fatty acid to form lipids. And this is also the essential component of cell membrane of cancer cell. So you will see that by having the glycolysis activity enhanced, you can also help the cancer cell to increase the structural molecule required for cancer cells. The extracellular pH, which contributed from the lactate or lactic acid secretion, also promote the cancer cell propagation as well through the key molecules, which is the extracellular matrix proteins. In fact, the acidic environment or pH promote the metastasis through the cytoskeleton remodeling. What we have here, we have the slide showing you on the left-hand side, which show you the normal epithelial cells. Then you have the transformed epithelial cells called the dysplasia, in which the junction of the cell is disintegrated and the cells lose the tight junction. In the end, this transformed cell become mesenchymal sort of type of cell where the adhesion between the cell completely lost, and that's resulting in the mesenchymal, in the transformation of the dysplasia becoming the mesenchymal cells, where the cell can start to migrate somewhere else because they lose all the tight junction and the adhesion molecules. Okay, and how the lactate or acid environment promote this process. Now, you look at the pH inside the cell and outside the cell. pH inside the cell we call pHI or intracellular pH. If you look at the pH value here inside the cell, roughly about 7.2. So this is quite neutral. And pHE or extra cellular pH, slightly alkaline, which is about 7.4. This is what's happening or what we found in the normal epithelial cells. There's a slight difference between inside and out the extracellular pH. Then when the cell become transformed or become the dysplasia, there's a slight difference between pH inside the cell and outside the cells. Yeah, you see here, the pH inside the cells are slightly above 7.2, but still perhaps not much comparing to what we observe in the normal cells. But what the drastic difference occurring outside the cell, where the pH drop from 7.4 to 6.7 in the extreme case, or 7.2, that's significantly lower than normal cells. Now, the drop of pH, in fact, can influence or can dictate the cell migration through the modification of one of the extracellular matrix molecules, which is the integrin. So you know that integrin is one of the key extracellular matrix proteins that kind of essential to hold the two epithelial cells together. Now, under the neutral pH, what they found is that the integrin protein is quite intact and can hold the interaction between two adjacent normal epithelial cells. So this is the drawing of the integrin. You will notice that the domain of the integrin, and this is the extracellular domains where it forms the head structure like this. So when the acid environment as a result of lactic acid secretion from cancer cell is high, you can imagine there'd be a high concentration of proton outside the cancer cells. And this proton, in fact, can protonate the amino acid residues, especially the histidine residues of the integrin molecules. And when the this key histidine become protonated, there is a conformational change, as you can see from the head structure. It used to be a very compact structure like this, now become open. And when the integrin become protonate, what will be happening is that it will activate the downstream kinase, which is called the FAK, F-A-K, SRC. FAK stands for the focal adhesion kinase. So this kinase, in fact, regulate the cytoskeleton movement through the downstream enzyme called the Rho GTPase enzymes. So when the FAK is activated as a result of the activate integrin, and this is of course a result from the acid environment, activate Rho GTPase, then these enzymes GTPase will regulate the cytoskeleton rearrangements. You compare with the normal cell, which is shown as a blue box on the left-hand side. When integrin is not activated, or other words, when the extracellular pH is about 7.2, integrin is in the inactive form, means that it cannot activate FAK kinase. When FAK kinase is inactivated, low GDP dependent kinase also inactivates, means that the cytoskeleton protein in the cell remain intact. What we're talking about, what are the cytoskeleton proteins? So we have the 4 major cytoskeleton proteins, including tubulins, actin, vimentin, and keratins. This is pretty much happening in the normal cell. They kind of form very tight interaction, and when they sit like this as a network, cell will stop moving. Okay, that's why the individual epithelial cell kind of grew together nicely. So as I said, as a result of the lactic acid secretion from cancer cell, when the pH is dropped, you have activation of integrin followed by activation of FAC and GGPS, and that can activate the movement or the protrusion of cytoskeleton proteins. As you can see here, one example is the vimentin. Used to be silenced in the normal cell. They can polymerize not only the vimentin but also the actin. You see that the red part here is now polymerized. You think about this molecule as a beads, when it's remodel as a so rigid molecule, it kind of like a chopstick, it's just pointing to the cell membrane, and that's promote the motility of the cell and make the cell start to detach, because you have this cytoskeleton molecule kind of gliding the cell to move outside the origins. That's when the process known as the epithelial mesenchymal transition is happening as a result of the extracellular pH change becoming more acid. So that explains why the acid environment promote cancer progressions. The acid environment outside the cancer cells kind of form the something known as the micro environment surround cancer cell. Otherwise, it's kind of form the shield from immune surveillance. You think about in our cell, when we have the tumor cell form, the T cell and other type of cell try to get rid of. You will see the antigen, right? Tumor antigen will come and destroy it. But in the cancer cell, they tend to escape the immune surveillance by forming the microacid microenvironment that kind of shield the tumor mass. So the when T cell coming in try to get rid of this tumor mass, they not work that well. This is because the acid environment, in fact, not good for immune cell in many ways. For example, cause for the antibody secretion. If you have acid environment, antibodies again, to destroy cancer cells tend to be degrade more quickly than under neutral conditions. You have the oxidation of the immunoglobulin molecules. It no longer work that well. There is the aggregation of the FC fraction, FC portion of immunoglobulins, make them become aggregated. The poor binding activity of the epitope on the antibody molecules to the antigen on the surface of tumor cells. Acid also downregulate the cytotoxic T cell and helper T cell, as well as you know that to be able to be able for the T cell to kill, you require the effective T cell or helper T cells. So when the pH drop, T cell doesn't work as well as before. So that's why the immune surveillance it drop. Dendritic cell also poorly work under this condition. And NK cell, which is function in killing specifically cancer cell, doesn't work that well because the low pH affect the this key molecule, for example, the cytotoxic activities and NKp46, which is one of the key molecule that NK cell used to kill cancer cell. The macrophage will function also impair as well. So that's kind of good for cancer cell by having this sort of low pH microenvironment that make them more resistant to immune destructions. So now you see that that's kind of the logic of what's happening, why the glycolysis become activated, and how the acid environment in fact promote cancer cell survival and progression. So what I have listed here are the major metabolic pathway which are readjust. The upward arrow means that the pathway is activated. And in fact, what we have today, we have the 6 lipogram pathway. We have the increased aerobic glycolysis, or other words, it's known as the enhanced Washburn effect. And this activation can be achieved through the by selectively activate 5 key molecules: glucose uptake, hexokinase, LDH enzymes, MCT transporter, and PKM2 expression. We also have the activation of pentose phosphate pathway, glutaminolysis, de novo lipogenesis, nucleotide synthesis, and the TCA cycloenzyme mutation. We'll go to one of each one by one. Okay, that's the evidence showing you that cancer cell, in fact, upregulate the glucose transport by increasing the expression of the GLUT1 transporter proteins. Now, what we have here is the classic, the PET scan for low deoxyglucose. This is one of the very early diagnostic of cancer patients in which the patient were administered the 4-low-deoxyglucose and this molecule of the glucose analog. When the cell tumor cell uptake 4-low-deoxyglucose, because this is the tracer molecules, you can see that when the cell accumulate glucose, you notice that there's be the appearance of the dark grain spot. Because it's the PET scan, the scan you throughout the bodies, you can localize which organ have deoxyglucose accumulates, means that you're very likely to have tumor in that particular area. Now, you see this list the different type of cancer patients. We have breast, we have esophageal cancer, lung cancer, colon cancer, and all sort of thing. You see that most cancer will accumulate glucose here, as shown by the dark green spots. And the final disposure of glucose will be happening in the urine or gallbladders. That's why all the patient display the final disposal of glucose for deoxyglucose. in the bladders, urinary bladders. But what the difference among cancer, you can see that there's the scatter of the fluorodeoxyglucose, means that this cancer cell uptake glucose more than normal tissues. That's what you learn from Wolfsburg. And the reason for why the cell increase the glucose uptake is because they upregulate the key transporter for glucose, known as the glucose transporter 1 isoform, or GLUT1. Now, what is the GLUT1? GLUT1 is the specific transporter for glucose. You see that to be able for the cell to further metabolize glucose to the glycolysis, you require the GLUT1 transporter proteins, which sit in the cell membranes. So what different in cancer cell? There's happen to be the massive upregulation of GLUT1 expression. This is to ensure that cell have enough glucose into the cells so that glycolysis can be upregulated. This is the evidence in the, this is the clinical data I'm showing you illustrated in the F figure here on the bottom right of the slide. There are well correlation between the percent survival of cancer patients who have higher level of GLUT1 expression, which is shown in the bottom graph here. Means that comparing with the cancer patient who have lower level of GLUT1 expression, you see that survival rate of patient whose tumor harbor higher level of GLUT1 have lower survival rates. See, whereas the another group of patients who have lower level of GLUT1 have longer survival times, means that GLUT1 can be used as the prognostic marker as well to predict the virulence or survival rate of cancer. By this tumor having higher GLUT1 is good for the cancer cell to progress, right? But then it's bad for us. That's why survival rates become this drop very rapidly. But the less aggressive tumor have less GLUT1, means that they progress lower. That's why we survive longer than the first group. Okay, and that's true if you look at the expression level of these proteins on the top panel here. We have the pair of tumor and normal tissues in 5 different patients. You will see that the GLUT1 level, in fact, it's very high in the tumor tissues compared to the normal counterpart, which is indicated by N or normal. You see that a lot less GLUT1. Compare with, similar with patient 2, 3, 4, and 5, they tend to have the higher GLUT1 in the tumor tissue than the normal tissue. And this is true also if you perform the immunohistochemistry staining of tumor tissue as well. You see that the tumor tissue tend to have higher or stronger positive staining for GLUT1 expression. This is telling you how elevated GLUT1 expression promote the aggressiveness of cancer cells. Okay, I think we'll have 5 minutes break, and they will come back. Okay, we'll come back again. Okay, that's the next one. Example is the increase of the key enzymes in the glycolysis, which is the hexokinase 2 enzymes. This enzyme regulate the catalyze the first step of glycolytic reactions, where glucose is phosphorylated by hexokinase to produce glucose 6-phosphate molecule. So again, you see the similar evidence where the malignant cell tend to overexpress the HK2 enzymes as indicated by brown staining here. We have two examples. One is the ovarian cancer. The malignant cell have more HK2 in the tumor areas, similar to the colorectal cancer cells, where the this enzyme is always expressed, as you can see by the very intense staining of the enzyme in tissues. In fact, what they also found is that HK2 in fact also serve as the anti-apoptotic proteins. So why the enzyme can serve the role as the anti-apoptotic proteins? Perhaps you look at the slide, the diagram on the right-hand side. We have two physiological situationsนะครับ. The first one where the glucose is very high นะฮะ other words cancer cell will use this condition to overcome resistance to apoptosis through the upregulation of HK2. Now, when glucose is high, glucose will enter into the cells. So under this condition, what they found is that the enzyme hexokinase 2 here, as indicated by this guy here, tend to bound close to the outer membrane of the mitochondria, or OMM is outer membrane of mitochondria นะฮะ so under glucose concentration that very high, what they found is that HK2 will form the trimeric complex with two proteins. First protein is the VDAC นะฮะ VDAC stand for the voltage dependent anion channel นะฮะ then the VDAC then also associate with the transporter, which is the ANT นะครับ ANT or adenine nucleotide transporter นะครับ so by having these three protein tightly associated they will form sort of physical barrier we will block the access of two apoptotic proteins. I hope you heard about these proteins. They are BAX and BAD protein. They are one of the key apoptotic protein which will tend to attack mitochondrial membranes นะฮะ Otherwise, under high glucose concentration, the apoptosis is inhibited. That's why we are saying in this sense HK2 serve as the anti-apoptotic protein because it form very tight interaction with VDAC and ANT. And these trimerics or tertiary complex block the entering of BAX and BAD to attack mitochondria. You know that if this mitochondria is attacked, the cytochrome C will be released and that's going to trigger the apoptosis. But if you deplete the glucose out, we see that now HK2 is released from the membrane and means that the trimeric complex no longer occur. And when HK2 dissociate from VDAC, that will allow BAC and BAD to access to VDAC. And they will start to attack the membrane, resulting the release of cytochrome C. And that's when the apoptotic is triggered. So, cancer cell knows that if they can upregulate glucose more into the cell through the HK2, then they will overcome apoptosis. And that's why upregulation of HK2 is essential for cancer survival. If you have, as long as you have HK2, then you block the apoptosis. LDHA overexpression cause LDH enzyme is on lactate dehydrogenase is the essential enzyme which will convert pyruvate to lactate นะครับ so LHA is very well support evidence that LDHA is overexpressed in most type of cancer means that they promote the Wasserburg effect ครับ LDHA also expression level is also correlated well with the resistance to the chemotherapy or radiotherapies means that any cancer cell which is resist to these two treatment will always or often have higher level of LDHA overexpressed นะครับ so in other words LDHA can be a prognostic marker for many type of cancer as well now in fact in human we have two set of genes นะครับ called LDHB and LDHA นะ they perform the similar reaction in terms of interconversion between lactate to pyruvate and of course the reverse reaction. Now there's the two isoforms, but if you look at the LDHB and LDHA, the native enzyme in fact is tetramerase proteins, means that you need the four subunits to work. For the LDHB, you can see that this LDH1 LDHB from the LDH1 homotetramer นะครับ LDH1 tend to favor the conversion of lactate to pyruvate more than the reverse reactions. So that's LDHB. Comparing with LDHA, which when you have four tetramer, they form LDH5. This is reaction totally opposite. This LDH5 will favor conversion of pyruvate to lactate more than reverse reaction. In other words, the kinetic property of LDH1 and 5 is quite different. LDH1 prefer conversion of lactate to pyruvate, whereas LDH5 or LDHA prefer opposite reaction. Now, in fact, as you see that there's a lot of pyruvate produced as a result of glycolysis. So that's explained that tumor cell will selectively activate LDHA expression so that it will produce LDH5 that will help in conversion of pyruvate to lactate นะครับ but think about it, if tumor cell selectively activate LDHB rather than LDHA, what would be happening? You would not be able to convert pyruvate to lactate, right? Because the reaction occur opposite way นะ means that they no longer convert pyruvate to lactate นะครับ that's the reason why tumor cell prefer LDHA นะครับ to be selectively activate นะ in MCT4 is the transporter which will help to secrete or export to secrete or export the lactate outside the cell. We call the MCT or monocarboxylic acid transporter 4. As you can see here from the cartoon here, by having both LDHA and MCT4, that will make the cancer cell able to secrete lactate outside of the cell, and that will create the acid microenvironment outside the cell. We have well evidence that MCT4 is overexpressed in many types of cancer. As you can see from this slide, this is the Western blot carried out into different breast cancer cells. The first one, HTB125, this is a low invasive breast tumor. Comparing with MDA231 cell, which is very, very aggressive tumor, you measuring MCT expression level, MDA, MB231 tend to have a lot of MCT4 overexpressed, and that can promote the virulence or aggressiveness of MDA, MB231 quite well. So by having MCT4, that can, help them to eliminate or secrete lactate outside the cell and make them survive well by having this MCT4. PKM2, this is the downstream enzymes in glycolysis which convert the phosphoenolpyruvate to pyruvate นะครับ the enzyme called pyruvate kinase นะ or PK นะ PK similar to LDH have multiple isoforms, but the form that will be selectively select to be activated in cancer cell tend to be the isoform called PKM2 นะ PKM2 also able to catalyze the conversion of phosphoenolpyruvate or PEP. This is almost the terminal glycolytic product ครับ By having PK expressed in the cell will be able to convert PEP to pyruvate so that glycolysis can be completed. But surprisingly about this specific PKM2 isoform is that they found that this enzyme in this isoform is very, very inactive in the way that the rate of conversion of PEP to pyruvate is very poor. So imagine if you have PKM2 with very poor enzymatic activity, what would be happening with this reaction? Means that reaction not very good, mean you end up with the accumulation of substrate, right, which is PEP. when you have the accumulation of PEP, means that they will drive the reverse direction of glycolysis, means that you end up with the accumulation of glucose 6-phosphate, which is the precursor for making macromolecule. So by having PKM2, they thought that by having poor activity, this will drive the glycolysis reverse, and that will favor the synthesis of these key biomolecules, which support the biosynthesis. Okay, that's what I just put in the slide. In the normal cell use PKM1, which is the very active glycolytic enzymes. So PKM1 will be convert phosphoryl pyruvate to pyruvate very well. So enough. But tumor cell, by having PKM2, which is very inactive, means that you block this step. That force the glycolysis to be reversed, and that favor the biosynthesis of the structural molecules. So apart from that, what they found is PKM2 perhaps not only play a role in metabolism, but it also play the what we call the non-enzymatic function นะครับ as the transcription factor or coactivator ครับ so now we have PKM นะ PKM2 can be phosphorylated by ERK2. This is one of the key MAP kinase pathway. Turn out what they found is that PKM2 act as a substrate or the target of ERK2 phosphorylation. ERK2 phosphorylate PKM2 at the serine 37. And once PKM is phosphorylated, what they found is that the enzyme can now, instead of localized in the cytoplasm, as a metabolic enzyme, you would expect enzyme have to be in the cytosolic location. But in fact, what they found is the phosphorylated form of PKM2 able to translocate to the nucleus, where it serves as the either transcription coactivators or transcription factor and bind to the target promoter นะฮะ for example the GLUT1 GLUT1 is the key transporter นะครับ so when it bind to the promoter you activate the transcription means that GLUT1 mRNA is more then protein will be more as well and that will support the cancer progression PKM2 can also interact with the beta catenins, and this is the protein essential in the mesenchymal EMT, epithelial mesenchymal transition as well. And that's when this pathway is activated, means cancer cell progress quite well. That's the benefit of having PKM2 acting as a poor enzyme isoform and also act as the non-metabolic function. This increased pentose phosphate pathway as well as I mentioned before, when you have high glycolytic activities, means that the glycolytic intermediate along will be also be very high, and that can be used to support the pentose phosphate pathway activities. As again, if you recall from basic metabolism, the pentose phosphate pathway is there to support the synthesis of ribose phosphate นะครับ This is the key molecule of nucleotide. And nucleotide is the building block of nucleic acid or genetic material of proliferating cells. So by having this, pentose phosphate pathway is activated, means that you help the cancer cell to make the new genetic material as well. It also produces NADPH, which is essential reducing equivalent which required to support the lipids biosynthesis. And in ADPH molecules also help to eliminate the free radical molecules or oxidative stress in the tumor cell as well. In other words, by having pentose phosphate activate, not only support the ribose phosphate synthesis, but also provide the NADPH. NADPH, as you see here, is one of the products of the pentose phosphate pathway. Again, you see similar evidence in the clinical samples where the key enzyme, I forgot to mention it. One of the first key enzyme of pentose phosphate pathway is the glucose 6-phosphate dehydrogenase or G6PD. We see nice evidence that cancer cells bearing high glucose 6-phosphate dehydrogenase or G6PD have the poor survival rate comparing with the cancer patient who have lower expression level of G6PD, as you see from survival plot here. Again, if you perform the immunohistochemistry on the top, you see that tumor cell would tend to have stronger expression of these enzymes. Similar for the Western blot in the different patients, number 1 to 4, within the tumor tissue always have G6PD very high compared to the normal adjacent normal tissues with almost undetectable G6PD level There's also cancer cell also not only use glucose but also use the glutamine amino acid to support the growth. And the reaction which involve the glutamine metabolisms we call the glutaminolysis So what is glutaminolysis? Glutaminolysis is the reaction which involves the breakdown of glutamine to glutamate. And then the carbon skeleton of glutamate then flow into the TCA cycle. Now we have two situations which differentiate between normal cell and hypoxic cancer cells. So you see normal cell, they two nutrient source. One is of course the glucose. The other one is glutamine. Glutamine, is amino acid. It have both carbon skeleton and amine group, whereas glucose only have the carbon. You see that most glucose, of course, are uptaken into the cell and metabolized to the glycolysis. And then you have pyruvate as the final product. Pyruvate enter mitochondria for TCA cycle, But look at the glutamine, relative amount of glutamine, as can be seen by the thickness of the arrow here, means that this have the lower arrow, means that normal cell tend to use a lot less glutamine than glucose. Comparing with cancer cell, you see that they much higher uptake of glucose and also glutamine, as you can see by how big of the arrow of glycolysis and glutaminolysis comparing with the left-hand side. So means that somehow in cancer cell, not only glucose is heavily consumed, but glutamine is also highly consumed. And this pathway is known as the glutaminolysis. You think about why the cancer cell require higher amount of glutamine. Now, in fact, this is linked to the metabolism of lipids. So you see that carbon skeleton from glucose flow into the cancer cell to pyruvate, right? And then for normal cell to make the lipids, you require the flow of carbon from pyruvate to the lipids. But in cancer cell, you know that the mitochondria metabolism is discontinued because most pyruvate convert to lactate, means that there will be a lot less carbon flow into TCA cycle. You think about lipids is the essential component of cancer cell. If the pyruvate here is 100% depleted towards lactic acid production, means that the non-carbon will be flow into TCA cycle. And you know that citrate is a precursor for lipid synthesis. If there's no carbon flow from pyruvate, means that you deplete the citrate, right? This means that there's no way cancer cell will be able to make lipids from glucose because this pyruvate keep diluting out as the lactate. The answer is cancer cell switch to use glutamine in the same time. They highly consume glutamine, and this glutamine convert into the alpha ketoglutarate, which is one of the TCA cycle intermediates, and that can be used to bypass citrate production and make the lipids eventually. This is the pathway of the glutaminolysis. Is the this is the fatty acid synthesis that you see. You require citrate, right? As I said, to be able to make the fatty acid as the key component of cell membrane, you need this pathway. And citrate coming from two sources. One can be from glucose, and one can be from glutamine. But because of the Wolfsburg effect, means that you deplete the flow of pyruvate into mitochondria, means that cancer cell have to use something else to provide carbon source. And in fact, that happen to be the glutamine. Glutamine will be incorporated into or uptake into the cancer cell and convert to glutamate by enzymes called the glutaminase or GLS. This is the reaction of glutaminase. You see glutamine have two amino acids. When one amine is eliminated as free ammonia, glutamine become glutamate. And then glutamate, as I point out here, further convert to alpha ketoglutarate by glutamate dehydrogenase or GLUT-D1. They will enable the cell to TCA cycle to flow again, even though you don't supply pyruvate into TCA cycle anymore. You can still get alpha ketoglutarate to flow as succinyl-CoA and the rest of TCA cycle until citrate is there. Now, citrate can then be used to produce fatty acids for cancer cell. So that's the explanation why cancer cell require a lot of glutamine. If you try to grow the cancer cell in the flask, under culture condition, if you happen to forget to add glutamine, you know that cell will not grow, because they require glutamine to support the fatty acid synthesis. What else we have? Cause the de novo lipogenesis, which is continued from glutaminolysis. You know that membrane lipids is the cellular structure of tumor. That's why there is no reason to believe that de novo fatty acid synthesis or de novo lipogenesis (DNL) has to be upregulated as well, so that to ensure cancer cell will have enough lipids. The two enzymes in this pathway, which can control the rate of lipid synthesis. First one is the acetyl-CoA carboxylase or ACC, and the second one is fatty acid synthase or FAS. These two enzymes working together. Here we have ACC catalyze the carboxylation of acetyl-CoA ซึ่ง derive from citrate to malonyl-CoA by having this enzyme upregulate cancer cell can increase the rate of fatty acid synthesis. Again, similar clinical association between level of ACC in cancer patients. So here we have example of hepatocellular carcinoma where the ACC1 enzyme is higher in the tumor area comparing with the adjacent normal tissues AC as shown by the immunohistochem, and again correlate well with the survival rates. So cancer patient who have the higher ACC would have shorter survival time than the those who have less ACC expressed in the tumor. Survival rate will be longer than the first group. So in other words, ACC expression can also be used as the prognostic marker to predict the outcome of the patient. Again, the for the synthesis of nucleotide also upregulate in cancer cell. They use the SSMT1 and 2 as a target. What is SSMT? SSMT stands for the serine hydroxymethyl transferase enzymes. This enzyme function in folate metabolism. So HSMT is form part of the enzyme that regenerate tetrahydrofolate. Now, you might find this a bit complicated, but in fact, the folate metabolism require SSMT1 enzyme to convert the interconvert tetrahydrofolate to methyl tetrahydrofolate. This part of pathway is essential to make DNA and RNA. If this pathway is impaired, cell no longer able to make enough nucleotide. Now, by again opposite, if you have HSMT1 and 2 upregulate, means that cell will be able to make a lot of DNA or nucleotide. And that's why in the cancer cells, they also overexpress SSMT1 and SSMT2. So basically, the two 1 and 2 they are isoform. 1 is the cytosolic enzymes, whereas 2 is the mitochondrial isoform. But they catalyze similar reactions in terms of regenerating the tetrahydrofolate. And we know that tetrahydrofolate is essential to make the uridine nucleotide. Uridine, is based in the RNA molecules. If we have more SSMT1 and 2, mean that cell will be very active in making nucleotide. Similar to the other metabolic enzymes, there's good correlation between higher SSMT and the poor survival of patient as shown by the immunohistochem. Pearly tumor or normal tissue area will have less SSMT. In this case, they look at SMT2 expression. Last one is the mutation of TCA cycle enzyme happening in certain type of cancer. Specifically, this one found in the glioblastoma of brain tumor. There's a mutation of enzyme in the TCA cycles called the IDH or isocitrate dehydrogenase นะครับ This enzyme normally um, have three isoform นะ IDH1 2 and 3 นะครับ In the mitochondria we have IDH2 and IDH3 นะ whereas in the cytosol we have the IDH1 enzyme IDH 1, 2, 3 differ in terms of cofactor utilization. As you can see here, the IDH1 and 2 use the NADP as the cofactor, whereas IDH3 นะครับ use NAD+. But the reaction in terms of driving product formation is identical นะครับ because they all convert isocitrate to alpha ketoglutarate molecules. Now, why mutation happening in this type of cancer นะครับ On the right hand side we have the hotspot mutation นะฮะ which indicate by this amino acid change for IDH1 often they found mutation in either IDH1 or IDH2 นะครับ The right hand side is the IDH arginine residues 132 is mutated to histidine or cysteine or glycine นะ lysine serine. This is the hotspot mutation for IDH1. Or they can be the mutation of arginine 140 นะครับ to falling residues นะครับ whatever mutation happening, what they found is that the mutants of IDH1 or IDH2 create the extra kinetic properties in the way that they can convert isocitrate to something else from what wild type does นะครับ For the wild type isocitrate should be converted to alpha ketoglutarate นะครับ Okay, that's what happening with normal cell. But for glioma with IDH1 mutants, what they found is that the isocitrate in fact can be converted to extra metabolite we call hydroxyglutarate นะครับ You say this is the diagram. Wild type you should be ended with alpha ketoglutarate. But for the mutants, they able to convert alpha ketoglutarate to hydroxyglutarate นะครับ This is we call oncometabolite. Now, there's a keyword, right? Prefix onco and metabolite means that this is the metabolite that's when created in the cell, it can transform normal cell to tumor cells. That's why we call it oncometabolite. Okay, that makes sense นะครับ oncometabolite can promote cancer in two ways because it can impair the normal reaction in the cell. First one, because the hydroxyglutarate 2HG นะครับ has a similar structure to oxoglutarate, means that it's create the inhibition นะครับ of reaction catalyzed by the lysine histone demethylase นะครับ and when this enzyme is inhibit นะฮะ because cell now see the 2HG as the substrate, it will block reaction. and that can affect the DNA methylation, causing epigenetic activation of some proteins. Or 2-HG can also act as the substrate for hypoxia activation. This is because for the hypoxia, you need the oxoglutarate, which should be the real substrate. But then when you have 2-HG in the system, means that this enzyme is confused. So it thought 2-HG is the substrate. So it means that you block the enzyme activity. This creates what we call the pseudo hypoxia, and it promotes cancer progression. This is the direct evidence. If you incubate the colon cancer cell SCT116 with the control, this is the metabolite they added to hydroxy glutarate. They to form DL and L form. You can see that, and then they do the look at the morphology. You see the cell that have the 2-HG tend to resemble the mesenchymal state. They used to be epithelial like this, but now when you add 2-HG in the culture media, cells start to become mesenchymal. Whereas the L form seem to be less potent than D form. Again, if you perform migration by wound healing assay, you will see that cell which exposed to D form of 2-HG can close the gap faster than control group. But again, L form is not very potent as the D form, so the gap close is lower. Similar for migration, invasion assay by transfer, you see that cell migrate to the extracellular matrix, which can be stained by crystal violet as the blue color. Cell are exposed to 2-HG deform, invade extracellular matrix better. That's why you see the strong staining of crystal violet. Stronger than L form. That's the direct evidence. Okay, having seen all the metabolic readjustment inside cancer cell, you might want to know what dictates this. In fact, the regulator of metabolic transformation happen to be the mitogenic signal by itself, the AKT signaling, hypoxia inducible factor 1, and SIIM6. They all three, acting in concert and transform the normal cell to tumor cell. Along the way, cell is forced to rewire or reprogram metabolism so that they will ensure there will be enough precursor to make the new structural component of the cancer cells. Okay, example is, as I said, the upstream event is the AKT signaling by growth factor overstimulation. You drive the signaling pathway. AKT is the most obvious kinase. And when it's promote, it can activate the downstream key molecules. For example, MYC. MYC, is one of the oncogenes. You have the HIP1 or hypoxia factor. And because these guys are pretty much the transcription factor, they can bind to the promoter of the target gene in metabolic pathway, some involving glucose uptake, glycolysis, nucleotide synthesis, and of course the lipid synthesis. HIP1 แล้วก็ MYC HIP1 is the hypoxia inducible factor แล้วก็ MYC work in concert That's the target of these two proteins you can see there's some overlapping targets. For example, glucose uptake, arrow mean activate นะครับ vertical line mean inhibit นะครับ inhibit ox force so forth with C นะ hypoxia นะครับ the hypoxic environment นะครับ in fact it activated by the hypoxia ครับ so if your cell have no oxygen นะครับ hypox the responsive protein is called the hypoxia inducible factor นะครับ or SIF นะฮะ so if this is the abbreviation is called HIF นะครับ or hypoxia inducible factor นะฮะ is the heterodimer proteins consisting of HIF1 and HIF2 alpha นะครับ they will recognize the responsive element called the hypoxia responsive element นะครับ or HRE นะครับ it respond to very low concentration of oxygen นะครับ or otherwise it support the growth of the solid tumor quite well. This is how HIP is activated นะครับ HIP is the transcription factor protein. Of course, this protein would display the signature domain, for example, the DNA binding domain นะอันนี้คือ N terminal นะครับ basic helix loop helix DNA binding domain นะมี dimerization domain. This is the what transcription factor look like. But what extra domain this protein has is called the oxygen dependent stabilization domain นะครับ make this protein can sense the oxygen นะ now look how oxygen can affect the function of HIP นะ this is the spherical drawing here representing this is the HIF molecules นะ there will be two critical amino acids which are proline and asparagine นะ so these two amino acid residues can be modified by reaction called hydroxylation นะ hydroxylation means you put OH group onto the amino acid. Now, proline can be hydroxylated. We call proline hydroxylation to become OH group attached. And this modification is catalyzed by enzyme called PHD นะครับ PHD stand for proline hydroxylase นะ who put OH group from oxygen นะฮะ onto proline. Okay, you have first hydroxylase, and then you have second enzyme called asparagine or asparagine hydroxylase นะครับ or FIH นะ functional similar to PHD นะ but target is now asparagine amino acid or ASN. When it hydroxylate, put OH group onto asparagine นะครับ Now, what happens when this HIP protein become hydroxylate? It will be inactivated นะครับ by proteolysis. Comparing with the unhydroxylate นะ Now we move to the right hand side with the HIP molecules without OH group attached to it. If the cell accumulate the non-hydroxylate HIP, you will stabilize its function นะ because when you have non-hydroxylate form, this non-hydroxylate form will recruit two additional protein นะ first one is P300. The other one is HIP1 beta นะฮะ when you have this trimer formation means that you stabilize and you enhance the transcription of HIP protein นะ Now move to reaction นะ hydroxylation on the right hand side. This is the poly residue on the HIP protein we talk about. And when you have proline hydroxylase, you will see that this reaction will require the input substrate. First one, the oxoglutarate. Second molecule is oxygen นะครับ other words under condition where oxygen is plenty นะครับ proline is 100% hydroxylate ถูกไหม because you have substrate which is oxygen. Remember when you have hydroxylate proline or hydroxylate HIP นะครับ you promote degradation. Okay, but if the substrate is not there, especially oxygen นะ so under hypoxia you have to think about tumor that grow deep into the vascular tissues with lack of oxygen, right? When cell lack of oxygen, this hydroxylation reaction does not occur, right? Because the substrate is oxygen is not there. Means that the proline is no longer hydroxylate. Means that HIP1 alpha is now in the non-hydroxylate form นะครับ now you move into the top one นะครับ this is trying to explain the connection นะ remember without oxygenใช่ไหม this is the non-hydroxylate HIP alpha will recruit the proteins will recruit the ubiquitination นะ ubiquitin you know is the protein tag with tag for proteolysis นะฮะ ก็คือ other words if you have non-hydroxylate as a result of the pathway protein degraded, but if you have the hydroxylation that will happen นะครับ the promote the because HIP1 alpha is the transcription factor when it stabilized นะครับ as a consequence of non-hydroxylation นะ it will enter the nucleus and bind to the target of several group genes นะฮะ angiogenesis erythropoiesis apoptosis นะ glucose metabolism itself ครับ okay that's a summary what I described for the HIP activation นะครับ so under normoxia นะ proteins become hydroxylated ครับ by these two enzymes and this will make enzyme prone to proteolysis นะครับ but if oxygen is very low นะ HIP1 alpha becomes stabilized and this bind to PT100 and CBP นะฮะ and that will then translocate into the nucleus and bind to the target enzyme นะครับ this is the target of HIP1 นะครับ not only angiogenesis but also glucose metabolism. So this is explain why under hypoxia condition นะ the glucose metabolism especially the glycolysis is activated นะครับ SIIM6 is also regulate metabolism especially the glutaminolysis นะอันนี้ energy production the glutaminolysis is known to be the target of SIIM6. Okay, as having seen how the reprogramming occurring in the cell, you might wonder if anyone ever target this key enzyme and use as the drug to treat the cancer นะครับ There is the commercial inhibitor right now target to the key enzymes in cancer metabolism นะครับ but they still under the preclinical trial and I think some of them work well and some don't นะครับ problem is because you think about the heterogeneity of tumor tissue นะ there's a mixed population of aerobic cell นะ some don't use the glycolysis นะ use phos but some for example the solid tumor นะครับ use glycolysis so the drug that target to the key enzyme in glycolysis might not work well with the aerobic cell. That's why they're still not very effective นะครับ because in fact the heterogeneity of cell in the tumor mass which have different type of metabolism นะ so I think it have to be ensure that there's if you select one enzyme it's going to work well for every type of population cancer cell population. Okay, I think that's come to the end of the lecture. I think you all have better. I know there's a lot of information, but I guess the key information is that going to be only in this slide. I think the first I get yeah, that's kind of the conclusion of the main metabolic readjustment. So you now see that by having this pathway activate how they benefit the cancer cell growth. Pretty much act by act through the key or regulatory enzyme that will enable the cell to make the essential structural component that support cancer cell growth. Okay, I think that's the end. Apologize for my late lecture ครับอาจารย์พิติ
ครับ
I just